Hello and welcome to the sixth of the biology videos. Today's topic is on ecosystems part 2. We're going to look at stratified sampling today. The purpose of it, the equipment or technique used, the method and how we gather data, how we interpret that data, and why we do this all in the first place. And it's to in turn be able to classify and better manage our ecosystems. Going to look at forests, soils, and reefs as well. So, this poster here has a compilation of images of many great scientists. The biologists include Darwin, Mendel, Louis Pasteur, Jane Goodall, Ian Wilson. But what ties all these scientists together is their ability to ask questions and to gather data. In ecology, it's unrealistic to be able to go out into an ecosystem and to just gather all the data there is on diversity, distribution, abundance of vegetation species. So we need to be able to split up a large ecosystem into smaller areas. This is called stratification. It can be horizontal levels or it can be based on zones of habitats such as elevation. This process of gathering data, sampling, based on smaller areas is called stratified sampling. Not only do we take samples representative of the whole, but we use the method of random sampling to minimize bias. This can be done using random number tables or various other methods. The technique or equipment used also varies depending on the purpose. So a quadrat may be useful when looking at immobile species, such as vegetation types in an area, whereas a transect might be useful when looking at the change of vegetation over a slope, being up a mountain or along the intertidal zone. And a plot is just the area under investigation. There's different types of transects as well, which we'll look at. So, transects are used often to create a profile of a terrain. A profile is the slope, position, and height, and shows different vegetation types and canopy cover. We also can create a plan sketch. So in a belt transect where we have not just a length, but also a width in which we gather data. A plan sketch is an aerial view of what we are looking at along that transect. So transects, if I flick back to this image, give us an idea of the vegetation or species over a large stretch of length. However, it would also be unreasonable to gather data all along the way. So if the technique is the transect, the method is the way we use that transect, and you might choose like every couple of meters to take a plot. Transects and quadrats can also be used together. To gather an idea of a population, the entire number of one species in a community, we can use this formula here. This formula refers to the capture and recapture method of sampling. And what it means is, at one point in time, a number of organisms within one species are captured, they're marked, released, and then at a later point in time, a second capture takes place. The point is to look at the number of marked individuals within that second capture. If it is a large population, then you would expect there to be a smaller proportion of marked individuals that reappear. But if it is a small population, then you would expect there to be a higher proportion of marked individuals to reappear. So this is the formula that is used, where M equals the number of originally marked, the first capture, N is the total number captured in the second sample, and lowercase m for Mary is the number of recaptured organisms that appear in the second sample. And this image here, I'll just talk to quickly before I go to the next one, refers to another type of transect. It's called a line intercept, and it would look at the number of species that are counted over a particular line, assuming there is a uniform, even spread of that population. A strip census, however, looks at the numbers in between the strip, just different ways of gathering data. So, a practice question. Here is a silverback gorilla. They are the largest primate, extremely strong. They can lift up to about 800 kilograms of weight, as opposed to a trained professional human, which can lift about 400 kilograms. To put that into perspective, a small car, such as a Micra, is about 1,200 kilograms. So they're incredibly strong. This question is referring to finding the number of mountain gorillas, such as a silverback, in a population using the Lincoln Index. 25, first capture, second capture, 24, and 8 marked were recaptured. Your job is to calculate using the index to find what is the number in that population. You can pause here, and you should have got an answer such as 75. Feel free to go over the working if you want to double check that. So, other types of ecosystems. Let's talk about soils and reefs. Soils are very important because they determine the type of vegetation, the flora that can grow, and that therefore affects and influences the biotic factors that may be present. Monocultures detrimentally affect soil composition because they take out biodiversity and actually disrupt the nitrogen levels in soil. Nitrogen is very important if you've ever worked with fertilizers or in agriculture. Cattle also have a detrimental effect because they compact the soil, therefore restricting growth. Farming tillage lifts and rotates soil, and this can cause water loss, dehydration. But some positive things that can increase soil health are plants. Plants create roots and anchors for soil, which minimizes erosion. Rotating monocultures with legumes, like vegetables, restore nitrogen levels. And also mulching and bark just prevents water loss. It's also important to talk about our reefs. Our reefs are not just beautiful and great for our economy, but they protect our land from winds and storms and other natural kind of disasters. However, they are under significant stress due to rising temperatures and increased acidity of the ocean. We've spoken about this mutualistic relationship that occurs, so I won't go into it again now. But the zooxanthellae algae are rejected from the coral in high temperatures. This causes the coral to die, leaving the coral bleached. Not only that, but the levels of acidity in the oceans are increasing. This is because of the increasing amounts of carbon in the atmosphere. The ocean then absorbs, and this increased carbon leads to the formation of carbonic acid, basically just making the water more acidic, and this carbonic acid interferes with the formation of coral, and corals are formed out of calcium carbonate. So, in terms of management, we study and we gather information to better manage and look after our ecosystems, and soils and reefs are quite important in the conversation of management at the moment. Not only that, but we also have old growth forests. Old growth forests are not just really old trees, even though that is one part of it. This is an image of what scientists think is the oldest tree in the world. It's in California, and they've come up with the year 4,700 years. And its name is Methuselah. We're talking about old forests though. And what makes an old forest an old forest, I'll get to this little guy in a sec. There's a few criteria. Basically, they haven't been disturbed by fire logging or grazing recently. They have an overstory, this region, that is relatively dominated with old large trees. They have a well-developed understory with shrubs and herbs, and they're quite stable in their composition. There's also a range of growing trees and dying trees, and this just provides many microhabitats, so a high biodiversity. And this is also why it's important to protect our forests, because we have so many different biotic factors involved. Managing old growth forests. The reason why we do this, and I'm going to skim it, but you can cover this in your textbook more thoroughly, is because when we gather data, we can better classify them and rank them based on what needs to be preserved or not. So old forests take a long time to grow. They have a high biodiversity and therefore should be preserved. And we're doing a pretty good job classifying these forests to reserve them. For example, but we also want to talk about using them, like creating paper and timber. So this is why we do all of that. In Australia, in southeast Queensland, 73% of all old growth forests in the region are protected. It's pretty good. Whereas in Western Australia, they're actually protected 100% against logging activities, and logging activities are restricted to new growth forests. A few questions for you now, based on what we've just talked about. This one refers to Lincoln Index and involves a couple of different samples. So I'll let you pause the video here and answer that. It's worth 2 marks. I'm including marks to show you that in these next few questions, I have cropped out the number of lines that the sample test has given you to answer, but when you see the more marks, obviously more is needed. Question 2, explain how the process of classifying ecosystems is an important step toward effective ecosystem management of old growth forests. The next one, 6 marks, getting up there. We're looking at the same area of land, pre-harvest and post-harvest, and it's asking you to identify an ecological surveying technique, so that's the equipment, and the method, how you will use that equipment to be able to gather information on species diversity between the two ecosystems, and to identify a strength and limitation of your equipment and of how you're going to use that equipment. I would suggest use more lines than this in your response as well. And lastly, a field investigation was conducted on a site in a rainforest, and this diagram shows that kind of forest. It asks you to describe the process of applying stratified sampling to assess this site. And a few tips here. In your answer, refer to the purpose of this assessment. So what are you trying to achieve by gathering data? What's the technique or equipment that you use? How will you use that equipment? Talk about how bias is going to be minimized. And then once you've got that data, what are you going to do with that? Maybe graph, how are you going to analyze that? Worth 6 marks, so that's a big question. There's quite a few points you could include in your response here. Pause the video, go through that. And lastly, I would like you guys this week to leave a comment. That can be something you either learned, were challenged by, or wonder. A question. Feel free to respond to each other's comments as well, and I'll go over them and respond to them likewise. To finish this video, I will leave you with a quote from Ian Wilson. This man has been called the father of biology, and as you might see, he's got a damaged eye. And just an interesting fact, it's because when he was a boy, he was fishing, he had an injury there, so he chose to study the small things in life, and he actually became the leading scientist in studying ants. Being the father of biodiversity, I think it's fitting to finish with a quote from him. And he says, "We should preserve every scrap of biodiversity as priceless while we learn to use it and come to understand what it means to humanity." Guys, God bless. I'll see you in class.